So in January of this year, NHGRI sponsored a 3-day workshop titled From Genome to Phenotype, Genomic Variation Identification, Association, and Function in Human Health and Disease. And this workshop was designed specifically to inform our strategic planning work for the 2020 plan. And Sharon Plon is one of the council members who attended that meeting, and we've asked her to give the report from that meeting.
Right. So good afternoon, everyone. And I am really representing a large team, as you'll see, who helped to put this together. And certainly during the discussion period, any of the council members or staff who were there, please join in. As you all know, we're in the midst of our strategic planning effort called NHGRI 2020. This is the website for it. And this meeting is just one of the many events that we've been holding for it. So this slide just summarizes some of our recent events. There was a training meeting in St. Louis. There was a meeting at the Informatics Conference, one at the Wellcome Trust, and at Biology of Genomes. And then you can see on the right that there continue to be upcoming meetings. So please, if you're interested in our planning efforts, please try to participate. So what I'm talking about is the Genome to Phenotype workshop and the rationale for it. It was held back in January. So of course, this is a component of the strategic plan. I do want to be clear, the major challenge was linking genome to phenotype. This was not focused specifically on genomic medicine. You'll see there's a lot of overlap, but we really tried to focus more on the research underlying understanding genome to phenotype. This was really the main workshop for the Division of Genomic Sciences related to the strategic plan, and obviously it's being integrated with the others. And it's particularly important to this group because many of the large flagship programs in the area of genomic sciences are turning over in 2020. So the strategic plan has a major impact on this area of NHGRI's effort. NHGRI likes pictures at workshops, you'll learn that. This was our picture. The pictures on the right are just to show you that lots of people participated. We had lots of sessions. There were 153 attendees. It was video casted and recorded. And in fact, people were tweeting away while we were doing it. And you can find anything you want to know or to watch on the website that's on the slides. So the goal of the workshop was fairly straightforward, which was to develop strategic approaches to find and characterize genomic variants. How do you link those variants with genomic elements? And then kind of the big question of how do you understand variant effects on human health and disease? And I would say, as you'll see, as I talk about the findings, there was a lot of discussion of the last one. And then, of course, our goal was to integrate across all these big ideas. So we were seeking bold ideas, and we told the attendees in advance about that. So projects, knowledge, capabilities. We were certainly looking to identify what new resources, methods, or technologies critical are still needed. Again, were there key collaborations, key groups that we should be working with in the future? And then really we were looking for opportunities that were unique for NHGRI. So not necessarily an idea that was very specifically related to one disease, but things that could really go across multiple diseases or multiple ideas that would be particularly suitable for NHGRI. And then the time frame, and that got talked about a lot, were what could we do in the next year? And people had very different ideas of what you could do in the next year. And then what kind of longer term projects, and sometimes whether you could do them in 10 years was a question. Okay, so the way we set up the meeting was unusual. We started out with setting the stage in terms of the vision. We actually had, I think, 3 people give kind of vision talks and then responses to each of those talks. Then really looked at the scientific issues. Where are we right now? What are the big challenges and what are the opportunities? And on day 1, we focused on variant discovery and association. And on day 2, functional element discovery, characterization, and how variants affect function. Although, to be honest, there were a lot of overlaps between day 1 and day 2 in ideas that were presented. And then, of course, the big question, what should NHGRI do? So the idea was to clarify, consolidate, and categorize ideas. That turned out to be a big challenge because there were so many ideas. And then kind of synthesize and prioritization. So we did actually do a couple of things to try to foster the idea generation. So we did poll for project ideas prior to the workshop, and I don't have the exact number. Mike might be able to comment afterwards, but we got lots of ideas. Then when everyone was sitting in their rooms, we had project ideas suggested during breakouts, and there were 6 different topics. So again, lots of ideas. And then the staff in particular and the program officers then really tried to group some of these ideas that were presented into themes. So I'm going to just give you some examples in these 3 slots. So first, in terms of variant disease association studies, here are some of the recurring themes. So increased diversity of participants was kind of heard loud and clear. Consider variant continuum from rare to common, not kind of separate into 2 groups, but really the whole continuum. A very powerful theme that emerged was genomic analysis from telomere to telomere, including structural variation and repeats, that we really should be able to read all the way across the chromosome. Polygenic risk scores, again, a big emphasis on applying them in diverse populations and various types of traits. Improve reference genomes, incorporating diversity. So again, the diversity theme. And then thinking about how we incorporate analyses and trying to combine them. So haplotype, gene by gene, and gene by environment effects. The next big theme that came out of it was functional characterization. So function, and people love to say things like this at the meeting, function of all genes, all regulatory elements, and all variants would be good. Engineered and natural genetic perturbations. And again, the term perturbation you'll see comes up multiple times. Great and interest in trying to do large-scale perturbation experiments. Phenotyping from molecular to organismal. And there was a lot of discussion about that. We're very good on the clinical side in phenotyping the individual. There are people that can phenotype cell lines, but we really don't know how to phenotype like organs. If you make a change in a mutation, how do we describe the change that might have on the kidney or the heart? Predictive modeling of genome function based on systematic data sets. Not to forget our ability to model in all the experimental data was kind of how one person put it. Test and model the role of cell-to-cell interactions in gene and variant function. So again, thinking about more complex systems than just cell lines or single-cell organisms. And then again, using modeling to determine which data types and biological systems should be invested in. So this was a very interesting idea of the modeling, not just on its own accord, but the modeling then to help determine what kind of experiments it makes sense to do. And then technology development. There was still a great desire for new technology, and people felt that this really represented NHGRI very well. Better sequencing, a lot of comments about better synthesis of nucleic acids, better functional assays of all sorts, improved single-cell and multi-omic assays and analyses, standards for evaluating the phenotypes that the other group thought were important, spatially resolved profiling of solid tissues. So again, this idea of can we do more work at the level of the organ, not just at the cell or the organism, and explore and develop modern machine learning methodologies. Okay, so then we tried to put all those ideas together and to clarify and consolidate the ideas, consider NHGRI's role, and organize the ideas. And so this was really the 3rd day. And although I did not start out as 4 topics, in the end, kind of organized the ideas into 4 topics. So in order to do the idea of prioritization, we actually did use an electronic polling method, that was used to rank ideas from the workshop. So my memory is they weren't tags, they weren't tickets. There was something like tickets. What? Dots, right, dots. So we each got, dot storming, right. It was a new term to me at the meeting. So we each got a certain number of dots, and then you could put dots on your favorite ideas, but there was some way in which then dots started to disappear. It was kind of a very interesting thing. And then as ideas got more dots, then you could try to reprioritize them. And those results were like immediately available. We were kind of watching the dots. It's important to realize that we had limited time. This was really the third day, and a lot of the discussion today and the discussion the staff's been having is how to take all those ideas and turn them into specific projects or concepts in the strategic plan. And it was hard to really compare high-level ideas in very different topics. So the following 4 slides was an attempt, I think a very good attempt, to try to present to you folks today the highest ranked ideas. So the first, with regard to variant discovery and interpretation, sequencing technology, single cell and multi-omic assays, and genome and epigenome modifying tools. So again, this idea that we really still need better tools. Functional genomics at scale, both at the cellular level and the model organism level, a big push for model organism work. Statistical and computational methods for data integration, networks, and models. Again, that's a very general statement, but I think really doing that with regard to genomic data and the new types of data that's coming out. Interoperability and usability of data resources. So this is somewhat relevant to our conversation this morning, for example, about ENCODE with regard to, that we were talking about within council, is how do we make the data that all these different projects are developing as interoperable as possible without requiring tons of work. And then again, this idea of perturbation. So perturbational and computorial functional genomics and microscopy at scale. And you'll see the word perturbation comes up a lot. The second big group was basic research anticipating clinical need. So again, deep perturbation profiling, if you could change every base, what could you get out of it? And then proximal molecular readouts of variants at every base. So this was the idea, again, of what are the best assays that might be biomarkers for important clinical need that you could use for these kinds of experiments. Single cell genotype and expression association EQTL projects. Improved data visualization tools for researchers, clinicians, and general public. This was an idea that really came up at the meeting, not in advance, that especially for the clinical community and actually for many individual laboratory communities, if we had better ways to visualize the data, again, the ENCODE example was given at the meeting, if there's 73 tracks or however many tracks there are, what are tools that make it easy for someone to find the track they want and visualize that data will make that data even more useful to the community. Single cell multi-omic technologies, both wet and dry lab, increase the computational biology workforce. So again, training came up quite a bit, even though it wasn't part of our mission statement, that we need to keep investing in training to have the workforce we need to really take advantage of these ideas. And then particularly from the clinical side, approaches to detect larger structural variants from materials that you might routinely get in a clinical setting, and to make those as cheap and as accessible as possible so that they can be used on large number of clinically important samples. The third group, and you'll see this overlaps a bit with the other one, functional consequences of genome variation, a complete atlas of all genes and their function, this group liked, this was all big things, a complete atlas of elements under selection or cis-regulatory elements and variants across cell types and conditions, and then having a computational model to interpret all of that data and visualize it. Again, this group of recommendations had a big emphasis on training opportunities in this area, and then high throughput measurement of variants in organisms and organoids. And then the final group of ideas, before we kind of open it up to some discussion, were what are the resources? This is something that obviously NHGRI has, really had a major role or major part of its accomplishments are really providing data resources to the rest of the research community and technologies. And so again, this idea that we really have to be able to do telomere to telomere assembly of human genomes and do that on a relatively routine basis. Large-scale perturbation in pools of cells followed by high content, high resolution data collection. And in many cases, this is again talking about doing things like functional assays or phenotypic assays after you do the perturbation. Building interacting data platforms. So how do we get our different types of data to talk to each other? So this is, variant portals and visualization platforms for large-scale genome processing, analysis, and integration, obviously probably via cloud computing. And then, again, trying to build transferable gene regulatory or multi-scale models from multi-layered data across biological systems. So if we develop something, can we develop it in a way that then can be transferable to other projects? And so the output were obviously, or the next steps really, are the report that was generated and I think is now available. Again, Mike Pazin and a large group of individuals from NHGRI, and I think they took out the slide that actually had all their names on it, put together a great report that some of us got to comment on with regard to near-term and longer-term ideas. Obviously, today is not the beginning, but part of our effort in the discussion to start integrating this into the final 2020 strategic plan. And then are there some of these ideas that we can actually implement now, even prior, or now or in the next year prior to the plan being finalized? And with that, the workshop materials are available again on the site, and you can watch the whole meeting. If you do that, I would suggest getting coffee. It was quite an active meeting. And then I see their slide is on here. Good. And then this was the strategic planning kind of expert group that helped put the day together. But really, particularly the people on the left, or my left at least, Eric Green, Carolyn Hutter, Elise Feingold, Adam Felsenfeld, Mike Pazin as well and many others actually did a ton of work putting this together. And that's our communication team. Thanks. So I tried to go, and I said about pictures. Here's all of us in the room. Oh, and the important part of this picture, I meant to say because this got tweeted, is that this picture was, there's two important parts on the person in red up at the podium, up at the front. The important part of this picture, and that's why I know when it was taken, is this was taken the last morning, like around noon. So it's just to point out that people really stayed and worked hard for the entire time. Thanks. So now I think we're going to have a discussion. Yes. And I know some people on the phone, I think, were at the meeting, and I know many of you were. So I would love to get some discussion if you felt that captured the ideas that you thought, or the ones that you thought were of the ones I said were most compelling to the group.
So why don't we do that? Let's start with the council members that were present. Jonathan, I know you were there. Do you have any?
I don't think I have anything to add. That was a great summary of everything. Yeah, thank you.
No, it was a great summary, Sharon. I'll just say one of the things I thought was particularly good was the diversity of participants that we had. We had a lot of younger people too, and I thought they made a great input.
Yeah. Yes, and I should say that for every session, if anything, we sometimes had trouble finding men, to be honest, for a few of the sessions, but we had a lot of women speakers. And that was a joke, but it is true. We had a lot of women speakers. And we invited postdocs, junior faculty, senior faculty. And really, we did get different ideas, I think, from the different groups. So it was great to have them all there.
Sharon, that was really interesting. I wasn't at the meeting, but I do have a question, and that is, that's a huge amount of work, and you started to allude to it a little bit, but could you have made that list up before the meeting happened? What was surprising? What was not at all surprising?
Well, I certainly couldn't have made that list up. Actually, I think what was useful were some, and I'm sorry, somehow I thought it was on an earlier version of the slide. The importance of evolution actually came up, and it came up that after, I think, the first 2 hours, the word evolution hadn't been spoken. So a couple of people got up at the microphone and said, why not? You know, how important it was. So that's why I feel a little bad that I think it was on one of the earlier slides. But I do think that some of the ideas that I wouldn't have predicted, like the data visualization, really just came out of the discussion. And then really the power of some of the ideas, the telomere to telomere idea actually was very forcefully made by a couple of the junior investigators actually at the meeting. And so maybe we could have made the list up, but I don't think we would have come up with the same kind of priority. Yeah. And I'm sorry, I don't know if anybody on the phone from council was there. I don't know if they were able to get on already or not. No? Okay.
That was, very exciting. One of the things, or two things that came to mind, one was quick wins. Or did you come up with a sense of what can be done quickly? The thing that I've been thinking about is the interoperability between Anvil and Data Stage. And, you have 140,000 genomes from NHLBI with the genomes from NHGRI, large number of phenotypes. You automatically have an opportunity to start doing this genome to phenotype work with a large amount of data. Certainly I've been in contact with the Data Stage people. I haven't, and they basically have said everything's going to be interoperable between Anvil and Data Stage, which I didn't know ahead of time, but that seems like one way of starting to move forward quickly. The other thing is question about going from cells to organs, and the question about at a certain point you have like a kidney in a blender, or you have pieces of the kidney coming out which may have different functions based on the SNPs that are there or whatever.
Well, I think there was a lot of optimism about organoids.
Mm-hmm.
And that, yes, it may not be a whole kidney, but that can we start doing variant analyses? And individual labs are certainly doing this very powerfully. But can we start thinking about systematic ways of, perturbing the genome and then studying, organoid development for the organ that's probably most relevant to that. So I think that's one way that people thought could be done in the nearer term.
Because certainly a lot of these things you can see going on for decades.
Right. Well, that's why I said the 5
so
to 10 year thing when you're talking every gene, every variant. I forgot what the third every was in that list being understood.
So, but was there a sense of this can be done quickly and this can be done in our children's lifetime?
Certainly, and I don't know, Adam or at least if any, because I think you folks have been thinking about that more, like which of these things might be done quickly versus not, or like, since they're the ones who fund things.
Yeah, I can just comment quickly. So, Steve, what you mentioned about interoperability between CCGG and top med data sets. Yeah, quick. So quick is a relative term. And this is something that there's essentially unanimous enthusiasm for because but it's still hard. There's a lot of details, but that is something quick. The other thing that I want to say is that there are actually some things I think some of these ideas in this workshop, we asked for, rather than quick things, we asked for things that were a little bit further out, this 5 to 10 year idea. And there's actually one idea, that, of course, that we already have a concept, and Eric mentioned, for genome references that at one point started to come up a little bit, and we said, you know, we're already doing that. Let's find something a little bit farther ahead.
But even that's a good example where if you think about the telomere to telomere, we should be doing that now. But we're not doing that on 100 samples or 1,000 samples. And to be useful for a lot of diseases, we're just going to have to be able to do it routinely. So that's an area where we might be working on it now, but then this kind of sets the stage for where do we want to be 5 years from now, you know, in that continuum.
Elise, do you have any? That's it.
Yeah, so I think it was a very good summary. Some of the things that stuck out to me were, again, that evolution thing,
Right.
and then the broad support for model organisms, and sort of related to what you were talking about. Model organisms have full organs,
Right.
and you can easily manipulate them. They have very short lifetime, so those things can be done fast, small genomes, quick and easy. So I think that's a great thing to work on function of the genome in the future.
Yeah, that's a great example.
So that was great, Sharon. You mentioned an emphasis on perturbation. I'd like to learn a little bit more about that. Was that to make hypotheses and test them? Was that to
No.
be comprehensive in the scope of what the variation we're looking at?
Yeah, I mean, it's a great question. I think there were two types of perturbations that were, well, more than two, but the two that will come to mind. One is simply CRISPR, essentially is a perturbation of the genome, and being able to use CRISPR, or obviously the many related techniques that have come out of it, to alter every RNA binding site, alter every base, and then what do you want to measure? So there's that. The other perturbation was the perturbation of taking variants we may already have, and how do we systematically look, for example, at environmental effects, right? How do we systematically, change some aspect of the environment and measure? So there were both ways in which the term perturbation was being used. But I think there was a lot of discussion about change every base and then measure something, change every amino acid, and then how sophisticated can the measurement then be? Was one of them. There was certainly a lot of discussion in the different groups about about functional assays, obviously, because we're talking genotype to phenotype. And then an issue that the group I was in, which was, how does this prepare us for clinical, was, just talking about the need for those functional assays to really relate to the disease pathology. That we often can do functional assays, but having those assays be predictive of pathology is an area that we're not as good at, and trying to come up with methods to do that, so that as we do these perturbations, we get outputs, we can really say that output is meaningful for disease.
So just to go back to something you highlighted earlier, I really was impressed by the number of young investigators, early stage investigators, postdocs, and early junior faculty, and some of the comments that they made. And I was very glad that they were so interactive. But that also highlighted one of the other things that came up over and over again, which was the training.
Right.
And the fact that we really need to have a strong training program, because so many of the individuals that are trying to use these data really don't have a good sense of one, how they were generated or what the impact really is. So I think the training part of it, because it did come up over and over again, I think was really important and something we need to pay attention to.
And ways to, as part of that, a couple of people commented on ways to keep people in the field. So the example that was given were trainees with computational sciences of all type backgrounds, and how good the market is for them in other spheres. And are there ways for us to keep it attractive to stay in genomics? Obviously, there were a lot of discussions about various starting salaries that we won't be able to meet. But are there other ways to try to keep that attractive, to keep people in the field who are graduating with those skills?
Jeff.
Yeah, Sharon, thanks for the presentation. Wondering about the human resource piece of this and whether we've got the patients and tissue resources to make all of this possible, research participants. You talked a bit about enhancing the diversity there, but is this going to require a whole new effort to recruit a large number of individuals with carefully defined phenotypes to make this possible?
Well, I think the answer to that probably is yes. There was some discussion about some of the current large cohorts and how those might feed in. There was an interesting discussion that Heidi Rehm and I actually participated in with is how much of having patients submit their own data, how practical is that? We've had some experience with that in ClinGen, and that really diagnostic laboratories have very, very large compendiums, but making it easier for the diagnostic labs to submit the data and perhaps then have participants add the phenotype on top of that. But certainly there will need to be large cohorts. There was obviously a discussion about how much the All of Us cohort might play into this, as well as other very large-scale cohorts that are going on around the world. Anyone back there?
Josh?
So this is maybe an interesting, maybe this is beside observation of what I noticed, but kind of looking through, it's interesting how, and I read the document as well, sort of how there's a lot of discussion about how the challenges around assessing phenotype and, as we talk about Anvil versus TopMed, not versus, but anding and interoperability, there isn't a lot of interoperability necessarily between the phenotypes, and they're carefully, sort of working through those and how we move forward. I was surprised at the sort of voted up recommendations that there weren't more direct references to how we could accelerate phenotype capture, either through or interoperability or those kinds of things. And do you think that's because of how far out they are, because of the audience?
I think it's more the participants. This and Terry. I know I saw Terry earlier. I'm not sure if she's. Oh, over there. Yes. So Terry may want to comment from the genome medicine series, where that's a frequently brought up topic, that this was not kind of the genome medicine group. And I think if we had been, that issue would have been brought to the fore much more than the people that were the participants of this workshop.
We are, of course, one big happy family.
Yes,
I know. But this one was focused really much more on the sort of basic science side of the house. We do have a meeting coming up in the fall that Anastasia is organizing in September that will be focusing specifically on things like the areas that you work in and genomic medicine in general.
Right, and that's where I think those issues will get addressed. I think this group recognized the problem. For example, we did not discuss a lot. We discussed briefly electronic health records and things like that. Really, the group of individuals where that's really going to be key and what they really work on will be more at the fall meeting.
So one thing that I'd maybe amplify, which relates to the perturbations, is that I think that we're very good at understanding now the links from genetic variation to genes in cis, or at least we know a lot of the molecular mechanisms through which that happens. But we're still not very good at understanding how those effects sort of perturbate through, flow through gene regulatory networks, and how those get transferred into translated into sort of organism level changes. And so I think that some of the themes that were touched on were kind of relating to systematic ways of measuring network changes, as well as you talked a little bit about sort of organ level assays and phenotyping that's sort of going beyond RNA and things like that.
Okay,
Thank you very much.
you're
Sharon.
welcome.
All right, that's it for our reports. We are on to something now called council-initiated discussion.